Here we begin chapter 18, part 1, a very, very timely chapter given the current events that are going on right now with COVID-19 infections and the significant percentage of patients who present with acute respiratory distress associated with that infection. Here, what I have in the picture on the left is the oxygen story. We all fundamentally understand that oxygen and carbon dioxide are transported in the blood and with the help of red blood cells. But I think that's sort of where our basic knowledge ends, and we need to take it to the next level. What I'm going to try to do in this lecture is to show you why we would pay attention to certain things in order to give you a clinical understanding of what's going on. It always comes back to the idea that the more we understand at the fundamental level, the more powerful we can be clinically. As I mentioned in the previous slide at the beginning there, what we're seeing in the current COVID-19 pandemic is the effort to try to sustain the patient's ability to transport gas to meet at least minimal metabolic need. This patient is unconscious, they are sedated, and they're on a ventilator here trying to sustain ventilation in the face of a diffusional challenge. That's fundamentally what's going on, that we are delivering gases at a rate and at a concentration designed to overcome the barriers of diffusion present during an acute lung infection. The inflammation, the buildup of fluids, both are very, very formidable challenges to diffusion. And so what we're trying to do fundamentally is deliver gas at the appropriate concentration and at the appropriate rate, not only to meet metabolic need, but to not poison the patients. Pure oxygen is toxic. That's the end-all be-all. Even now, when I was first training, there was a debate about giving pure oxygen to neonatal patients, particularly preemies and so forth, because the pure oxygen causes lung damage. It is, in fact, a reactive oxygen species, just like other molecules are. And if you go scuba diving with a tank of pure oxygen, if you go down below more than 20 or 30 feet, you're going to die very rapidly from oxygen toxicity. Oxygen is a toxin. We deliver oxygen or try to deliver oxygen in a homeostatic window. But if the barriers of diffusion, the barriers to diffusion are so great, then we have to manipulate those variables. And that's what's going on with these ventilators. That's the whole story, is how do we get gases into and out of the patient in a way that isn't causing further damage if possible. Trying to save their life first and foremost. If we have to give them pure oxygen, we will. But if we don't have to give them pure oxygen, they're certainly going to try very, very hard to deliver what's needed. And so I have this figure here for you. And again, we all sort of understand this intrinsically. We ventilate, and that was the point of chapter 17. And why do we ventilate? In order to sustain diffusion gradients. We want to keep oxygen somewhere around 100 millimeters of mercury. And remember that pressure, that's a partial pressure, is synonymous with concentration. We also want to keep CO2 levels low enough that we facilitate unidirectional of carbon dioxide out of the blood. That's where if we don't breathe enough or if we breathe too much, we can screw up these ratios and lead to issues downstream from there. If we breathe out too much carbon dioxide, we start losing pH control, among other things. If we don't have enough oxygen, then we don't meet metabolic need. And so we're going to talk about these mechanisms. We're going to talk about the oxygen mechanism, and we're going to talk about the carbon dioxide mechanism. Both here at step 1, okay, step 1/step 6, as well as step 3 and step 4. So this chapter is fundamentally about this here at 1 and 6 at the level of the alveolus, and at 3 and 4 at the level of the tissue. It's the tale of two locations in the body. I was going to say a tale of two cities, but it's the tale of two locations in the body, at the tissues and at the lungs. But the question here is, How does this work? We know what's going on, that we're breathing in order to sustain oxygen diffusion. We're breathing in order to sustain carbon dioxide diffusion, not only right here, but also down here at the tissues. How does this work? We're going to go into some of those chemical details and solubility details. Why do we have hemoglobin in the first place? Okay. Gradients must be sustained. Okay, we have to sustain gradients here. We have to sustain gradients here. The membranes must allow diffusion, and that's the fundamental issue right now with COVID-19, is that we have barriers to diffusion at the level of the alveolus. Thirdly, we have to think about solubility. Why go to all the trouble to make hemoglobin if the gases are soluble enough to travel on their own through the bloodstream? That's the story of oxygen primarily. But the blood does more than just carry oxygen. It actually facilitates the transport of carbon dioxide, but it doesn't necessarily transport the majority of it. That's the interesting story of solubility in chemistry, both in terms of oxygen as well as carbon dioxide. Just for a moment, let's circle back for just a second to Unit 1 and a little bit of Unit 2 and 3. The idea that we're not using membrane carriers to move gases. We are actually using diffusion. That is to say, we're sustaining gradients in order for diffusion to occur. We're not using any type of facilitated diffusion in this case. And so not only are the basic rules of diffusion in place, but also fixed law: surface area, thin membranes, make sure that we have an adequate concentration gradient, and that the membranes are permeable to the molecule. Some of that depends on the solubility, and certainly CO2 and O2 are plenty soluble to move through membranes. They're small. In the case of oxygen, relatively lipophilic. Okay, so all of these things are at play, and we're going to show you some examples of where this diffusional barrier is disturbed in certain instances in this lecture. I'm certain that we all intrinsically understand the implications of hypoxia, but I'd like for just a moment to dig a little bit deeper into hypoxia, give you just a brief definition, and get you thinking about hypoxia in a little bit different way other than that sort of life or death thing. Hypoxia at several levels, it doesn't have to be absolute, is clinically relevant in any number of types of conditions. There are conditions that will contribute to hypoxia. There are conditions that will be affected by hypoxia. So what is hypoxia? Hypoxia is either insufficient oxygen to meet metabolic need in the tissues or the body, or the inability to metabolize oxygen in a tissue or the body. Two different things there, and we can actually break out different forms of hypoxia. We'll do that in just a few minutes. But as examples of things that either will contribute to hypoxia or are affected by hypoxia, certainly heart and neural tissue and kidneys are all sort of beholden to oxygen to make ATP and to function under normal conditions. They can't metabolize glucose without oxygen. They really can't do anaerobic metabolism. They don't have a sense of humor, according to one surgeon that I talked about. But certainly things like renal disease are affected by hypoxia and can drive forms of hypoxia. Atherosclerosis, we talked about the forming of collaterals, but it would also be an issue when we come to myocardial infarction and heart failure can interact hypoxia. Rheumatoid and chronic arthritis. Anytime we have inflammation in tissues, you're altering the fundamental flow of blood to and from that tissue, which can and does drive hypoxia, leading to secondary situations. We call those sequelae. Inflammation can actually be not helpful. If you get inflammation in the wrong place in the brain, it can lead to hypoxia and otherwise uninjured tissue. Certainly cancer and therapies trying to trying to slow tumor growth and so forth involve hypoxia. There's a lot to think about when it comes to hypoxia because it's about not only delivering oxygen to meet metabolic need, but also the ability or the requirement to metabolize oxygen to make ATP. Another thing I'd like you to think about in this chapter as well is this term called hypercapnia. Oftentimes we think of oxygen and carbon dioxide as being sort of yin and yang. When we have lots of oxygen, we have low carbon dioxide and vice versa. And that's often true. That's often true because we're using similar mechanisms to lower carbon dioxide out of the blood and raise oxygen in the blood. But you can have simultaneous oxygenation and too much CO2. If you ever see the movie, to extend a little bit on the idea of hypoxia, I want you to look at the four major classifications of hypoxia. The first one is called hypoxic hypoxia. That is to say, you can't, for whatever reason, oxygenate the blood. There's either too little concentration of O2 that you're breathing, or you've stopped breathing, or there's some sort of something going on, and you're low arterial PO2. This can be due to low partial pressure of oxygen, like in the case of high altitude, or if there's an oxygen depletion event. If somebody's in a place where there's low oxygen concentrations, that could be an issue. High altitude is probably the most common situation of this, or accidental suffocation in confined spaces. Where if we stop breathing for whatever reason, alveolar hypoventilation, or there's an obstruction of some kind that blocks. In the case of choking would be an example of alveolar hypoventilation. The inability to diffuse because of barriers to diffusion. This is really what's going on in our COVID-19 cases, is that the barriers to diffusion have expanded, or we have some issue with VQ, something along those lines, where we have an altered ventilation perfusion ratio. We have a type of hypoxia called anemic hypoxia, where for whatever reason the O2 cannot bind to hemoglobin properly. This can be for reasons of blood loss or low concentrations of red blood cells or low hemoglobin within those red blood cells. We call that hypochromic anemia when we don't have enough hemoglobin. Or there's something wrong with the hemoglobin, either because it's malformed, iron deprivation. Carbon monoxide poisoning is an example of anemic hypoxia. Why? Because carbon monoxide is a competitive inhibitor of oxygen binding hemoglobin. It has higher affinity for hemoglobin, and so will displace hemoglobin or oxygen from hemoglobin, and thus you don't have as much bound. Then there is ischemic hypoxia. This is fundamentally Q. For instance, you could have systemic whole body hypoxia due to the heart not beating, either not beating enough or not beating at all. You can have issues of shock where you have vasodilation, blockage or occlusion in the case of either like a thrombus or an embolus of some kind. Even inflammation can drive ischemic hypoxia. When you inflame and vasodilate and alter the fluid dynamics, you can increase tissue fluid pressure, and that causes pinching of blood flow in an area. Reduced blood flow can lead to hypoxia in that organ. This is what happens when you have hematomas, like for instance in the brain. What will happen is the increased fluid pressure will occlude the capillaries, and now tissue that's normally fine and healthy and uninjured no longer has enough blood flow, and you start to see secondary injury. And then the last one is called histotoxic hypoxia. And this feeds into my statement about either we don't have enough oxygen to meet metabolic need, or we cannot metabolize that oxygen. And histotoxic hypoxia is a failure of the metabolism. The cells cannot use the oxygen because they've been poisoned in one way or another. The most common instance, or the easiest one, is cyanide poisoning, which uncouples the citric acid cycle from electron transport. So I wouldn't say patients, but when folks have been subjected to cyanide as part of capital punishment, one of the things that you see is their temperature spikes because what happens is their citric acid cycle goes into overdrive because of the lack of ATP. And that's what's going on is that the cyanide prevents the use of oxygen to make ATP. And so the body is compensating by trying to make more electron transport molecules. Because hypoxia and hypercapnia are so very, very important to moment homeostasis, the body actually has overlapping windows, homeostatic windows that it works to control. So overlapping redundant systems in order to keep three different variables within that homeostatic window. Yes, they are related. Yes, they do influence each other. However, they are and can be mutually exclusive. That is to say, you can have lots and lots of CO2 even if you have plenty of oxygen. You can have plenty of oxygen but have issues with pH and so forth. So yes, they are overlapping. That's because we use similar mechanisms to manage them, but they are not necessarily dependent on one another. Let's just go through an example here with this figure here in the middle of the photo. Let's say we're working out. We're doing lots and lots of metabolic activity. Well, or we have a fever. That's another example of that happening. Well, we get increased metabolic byproducts, which include things like lactic acids and other forms of organic acids as metabolism occurs. This will cause an increase in the concentration of protons. And an increase in H plus means a decrease in pH. An increase in H is a decrease in pH. The more concentrated the protons are, the lower the pH is. If we have deviations of oxygen, carbon dioxide, or pH, they will stimulate both central and sometimes peripheral chemoreceptors that will then send signals to places like the pulmonary control center in the medulla that controls rate and depth of breathing and alters the rate and depth of breathing in order to either increase or decrease alveolar ventilation, either in terms of rate or depth or both. If we have more acidosis, we're going to increase rate and depth. If we have more alkalosis, a decrease in pH, we may want to interrupt breathing and slow depth. That's the point of hiccups. If you ever get hiccups because you're hyperventilating, like from when you're laughing too hard, what's happening is that your chemoreceptors are picking up deviations in these variables, sending a signal to trigger diaphragmatic convulsions, diaphragmatic spasms, which are hiccups. But we control these things very, very closely. Remember our pH window, somewhere between 7.35 and 4.5. Remember that 100 millimeters of mercury I told you about for that magic number? Well, I've got one more for you here, and that's 40 millimeters of mercury in terms of the arterial blood. So 140 and 7.4 are sort of our magic numbers for all three of these. This homeostatic window, 35 to 45, 85 to 100, 35 to 45 for our purposes. That's fine. But in any case, those are the three magic numbers now that we want to see in the arterial blood. Venous blood's a little bit different. Why? Because we're carrying more CO2, less oxygen, and so the pH is going to be a little bit lower. So here are some more examples of what would cause hypoxic hypoxia. Inspired air with low oxygen partial pressure. Content, partial pressure is the key. 100 millimeters of mercury. So low partial pressure of oxygen can drive low PO2. Altitude, so if you go to a higher altitude where your overall atmospheric pressure drops, the partial pressure of oxygen also drops. Okay. We have 21% oxygen at the top of Mount Everest as we do at the bottom of the Dead Sea, the lowest point on Earth. Alveolar ventilation is inadequate. That can be because of issues where you can't expand the lungs properly, or we have increased airway resistance because of inflammation and or occlusion. or we have issues with the breathing center in the brain being depressed, and so we're no longer breathing. Alcohol poisoning is very, very common. Drug overdose. This is part of the opioid crisis that's going on, is that drug overdoses are suppressing the breathing center, and so you literally stop breathing. And if nobody's there, you suffocate. So a little bit of review from chapter 17. Remember, we have to have a very, very thin layer right here, usually 1/10 of a micron up to 1.5 microns, in order for the rate of diffusion to be normal, in order for us to achieve appropriate oxygen saturation in the hemoglobins and achieve proper release of CO2 back into the alveolar airspace. So when we're managing rate of diffusion, we not only have to deal with the diffusion laws, but we also have to overcome the solubility issues, the physics, if you will, of the gases that we're trying to control. Some of the normal constants, these are constants in a moment-to-moment basis, but these things can change if we have long-term pathophysiology. For instance, surface area. If surface area declines, we lose the ability to achieve diffusion. Membrane thickness, whether we're talking about fibrosis or buildup of material on the inner surface of the alveolar membranes, that can fundamentally influence diffusion. Or diffusion distance, that may be a function of fluid either inside or outside the alveolus that separates the alveolar membrane from the blood plasma. So if we alter any of these are normally constants. We don't see these change very, very rapidly. Maybe diffusional distance under extreme circumstances, like in the case of near drownings or acute respiratory distress. Concentration gradient is critical. Ventilation and proper perfusion are critical. If we don't have those, we don't have appropriate gradients to achieve diffusion. That's the primary factor, and that's why we control ventilation and cardiac output so from second to second, moment to moment. Even just the act of getting up out of your chair, you can feel your heart rate change. So what changes in pathology? What happens when we have these pathological changes? Lose surface area. And the classic example of this is emphysema, where we lose surface area. We maintain total three-dimensional lung volume, if you will, but we lose a lot of the membranes inside the lungs, and that leads to alveolar surface area loss. There's also fibrosis with emphysema, but we're just going to leave that where that is. Fibrotic lung diseases, things like asbestosis or pulmonary fibrosis for whatever reason. Like for instance, we're seeing a raging comeback in pulmonary fibrosis in coal miners. Why is that? It's not because they're breathing more coal dust. Turns out they're breathing more other silicates because the coal veins are no longer as rich and thick as they used to be. So the coal mining machines are now cutting more limestone and more other stones rather than coal, and that's causing an increase in silicosis, leading to fibrotic lung disease. Pulmonary edema, surfactant disturbances are both examples of where we increase diffusion distance, either because there's more fluid inside the alveolus or there's more fluid outside the alveolus between the the capillary and the alveolar surface. So surfactants can do that, pulmonary edema can do that. But these all come back to Fick's law. Surface area, and then the basic rules of diffusion, gradient, temperature, etc., etc. So here's an example, as I mentioned in the previous slide. Emphysema leads to, the condition of emphysema is the result of destruction of alveolar surface area. So there's less surface area for gas exchange. Over here on the right would be a normal cartoon alveolus, and you can see there are more pouches, more membrane surface, and when there's more membrane surface, there's more opportunity for gas molecules to interact. Fibrotic lung disease, like I mentioned in the coal miner situation, there's increased thickness of membrane, that's increased diffusional distance, loss of lung compliance, loss of the ability to stretch. That leads to less diffusion, leading to hypoxia in the arterial blood. And again, here's a normal alveolus over here on the right to show you the difference. Pulmonary edema can either be fluid inside the alveolus or outside the alveolus. It really doesn't matter. What we're talking about is increased diffusion distance, which then leads to decreased difference, specifically of oxygen. The reason being is that oxygen has a lower solubility than CO2 in water. CO2 has a much higher solubility in water, so it's not nearly as affected as oxygen is, because oxygen is not as nearly as hydrophilic as water. And again, here's your normal alveolus on the right. Pneumonia or pulmonary edema are both examples of this issue. When it comes to asthma, generally what's occurring is that the inflammation and the secretion of fluid and mucin in the airways leads to increased resistance, and so you have alveolar hypoventilation. So low oxygen means that we have less diffusional. CO2 can be screwed up as well, particularly in acute cases. As opposed to normal situations, you can look at the diameter here. The bronchioles are constricted, increased resistance, less flow. It's always back to that basic rules of diffusion. Getting oxygen to move unidirectionally in, CO2 to move unidirectionally out. If you alter ventilation, if you alter barriers and distance, then you have issues with appropriate delivery of oxygen. So pressure gradients are important. Trying to sustain 100 millimeters of mercury of partial pressure of O2. Trying to sustain 40 millimeters of mercury of CO2 so that we have the appropriate pressure gradients. Okay. Solubility we're going to talk about here in a couple of seconds, and that's the story of hemoglobin. Temperature can play a role as well. I'll talk about that a little bit more when we talk about the ability of O2 to bind hemoglobin. That's where most of the temperature issues are present. So we'll get to that here in a little bit. So what's the big deal about solubility? I mean, fish seem to be able to do okay, and amphibians can seem to live underwater and diffuse gases, and oxygen seems to be present in water, or those animals would die, right? Well, there's more to it than that. As high energy organisms, we have higher metabolic rates than fish or amphibians or most aquatic organisms. Oxygen solubility becomes very, very important. And so let's go through a comparison of solubility, if you will. And we're going to do just a couple of quick experiments. On the left here, we have a beaker full of deoxygenated water. That is to say, there is zero oxygen right now. So there's zero quote unquote pressure in that water. And if we have 100 millimeters of mercury of oxygen in the atmosphere above that water, let's say this is a sealed container, we have 100 millimeters partial pressure here, and we have zero here for now. As we let that sit, it will start to equilibrate. We have a gradient, right? High, low. The water is going to start dissolving, or the oxygen is going to start dissolving into the water. Just like any other dissolved substance will. It will begin to diffuse down its concentration gradient. What will happen eventually? We'll reach equilibrium, right? Where the rate of oxygen moving out of the water is the same as the rate of the oxygen moving into the water. And so what do we have when we have equilibrium? And let's presume for a moment that we have unlimited amounts of oxygen. It's always going to be 100 millimeters of mercury. We're not equilibrating between these two compartments. Let's just say that that's 100 millimeters. Well, when we equilibrate, we're going to be at 100 here and 100 here. But notice something. The actual concentration in terms of millimoles per liter is much lower than in the atmosphere. In the atmosphere, it's about 5.2 millimoles. In the water, it's about 100 or 0.15 millimoles. That's about 3%. So at identical pressures, when we have achieved equilibrium, The 100 millimeters of mercury in air is equal to that millimolarity. And 100 millimeters of mercury in water, when it's achieved equilibrium, is 0.15, 3% of the atmospheric oxygen. So even though they have same pressure, because they're at equilibrium, they have different concentrations. That's solubility, and that becomes a life or death thing. So 0.15, if you take 0.15 and divide it by 0.520, you get a number of 3%, 0.03. If you multiply 5.2 times 0.03, you'll get 0.15. Okay? So even though they have identical pressures, here's 100 millimeters of mercury, 100 millimeters of mercury, they're in equilibrium, we're losing oxygen as fast as we're gaining it, we still only see 3% of the overall molarity that we see in the atmosphere. In contrast, if we look at carbon dioxide, and again, they obey the partial or the ideal gas law. So 100 millimeters of mercury in the atmosphere of CO2 would be 5.2, just like oxygen, the same number of molecules of CO2 per liter if we had 100 millimeters of mercury of CO2. But if we look at 100 millimeters of mercury of CO2 in water, the molarity is 3 millimolar. Remember that oxygen was 0.15, and this is 3. This is a 20-fold increase in solubility of CO2 over oxygen. Not quite 20-fold, 58 versus 3. Okay, so CO2 is much, much more soluble. It said nearly 20-fold more soluble in water at identical pressures and in equilibrium than oxygen is. Given that complex multicellular organisms, the vast majority of complex multicellular organisms have a metabolic rate that is too high to be sustained by the pure solubility of oxygen, evolution has provided us a workaround. And that is a carrier molecule, a carrier protein that we sequester inside red blood cells known as hemoglobin. Turns out there's a close cousin of this in muscle fibers called myoglobin. It is fundamentally an oxygen carrying molecule. It helps concentrate oxygen to meet metabolic need in organisms that get their oxygen from water directly. And it helps us to overcome solubility issues from animals that garner their oxygen from atmospheric oxygen. Because oxygen is so low soluble, we either have to concentrate it or we have to overcome that low solubility in the blood plasma. And that's what the hemoglobin does. This here, the first thing that happens is oxygen diffuses into the low concentration of oxygen in the blood that has come from the heart. So when the blood enters the lungs, it has a low concentration. It's somewhere around 46 millimeters of mercury. We want to get it to 100. So law of mass action. Oxygen starts diffusing into the blood plasma. But that's not the end of the story. Oxygen will also diffuse into the red blood cell and bind with hemoglobin, forming oxyhemoglobin. So we're taking mass away from this equilibrium, and so more oxygen comes in here, more oxygen comes to meet the loss of equilibrium here, and so we have a sort of unidirectional flow of oxygen when we are in the lungs. So we saturate the soluble oxygen in the blood plasma. We saturate the hemoglobins close to as much as possible. That gives us about 98% oxygen and somewhere between 2 and 3% oxygen that stays dissolved. But this right here and this right here and even this right here to a certain degree is law of mass action. Okay? A plus B, AB. Okay, that's the same story that we've talked about all semester. Now this red blood cell and plasma travels from the lungs to the tissues, so we transport it to the tissues in the systemic circulation, and we have the reverse going on. We have a diminishing concentration of oxygen inside the cells as it's being metabolized. Well, the oxygen that's dissolved in the blood plasma will diffuse in, again, law of mass action between here and here. As oxygen drops here, oxygen leaves the red blood cell, law of mass action yet again. So we have the oxygen dissociating from hemoglobin to replace the dissolved oxygen in the blood plasma to replace that which is being metabolized. And that's the story of oxygen in the systemic and pulmonary circulation. About 98% is carried as in the form of oxyhemoglobin. 2 to 3% or so is dissolved, whether it's in the blood plasma or in the inside the cytosol unbound hemoglobin. Since we're pushing about 35 minutes now, I'm going to go ahead and stop at this slide. We're going to talk about these different factors when we pick up with Chapter 18, Part 2 in the next lecture. Thank you very much for your time, and I will talk to you soon.